Hello, everyone. So today we're moving on to talk about experiment number 5, which is titled Extraction of DNA Molecules. So what we're going to be doing today is we're going to be learning how to extract DNA molecules from cells, specifically nucleated cells. So we're going to take blood and then we're going to isolate DNA from blood. And as you guys know, the DNA will come from white blood cells, which are the only nucleated cells in the blood. Okay. So to do that, we have to break the cell membrane open. We have to break the cell membrane open, and we have to break also the nuclear envelope. So we have 4 objectives for today. The first objective is to define DNA and its function, which you guys have pretty much covered in the theory part, in the lecture part. The second is to describe DNA structure. This was also covered in the theory part, but we'll go over it quickly. And the third goal for today is to understand the principle of DNA extraction. How does it work? What is used to extract DNA? What is the background theory behind it? And to efficiently extract DNA. How can we do that efficiently? The fourth is to list factors that may negatively affect DNA quality. So factors, environmental, for example, that can affect the quality of the DNA that we obtain at the end of the extraction. Now, what are nucleic acids? Nucleic acids, they are biological macromolecules, okay, that is passed from one generation to the other. It specifies information, for example, hair color, eye color, etc. So they are the hereditary determinants of living organisms. They define traits. Now, nucleic acids, they are polymers. They are polymers of repeated building blocks, and you guys have learned this already. So these small building blocks, these monomers, are called nucleotides. There are 2 types of nucleic acids, as you guys have already learned, DNA and RNA. So DNA and RNA. What is DNA? As I said before, DNA is a polynucleotide that contains instructions. Instructions for us and for other living organisms to develop, survive, grow, and reproduce. Now, you guys have learned already about the central dogma, which is a 2-step process where we move from genes to a protein. So DNA to messenger RNA, that's called transcription, and messenger RNA to proteins, this is called translation. This is the central dogma of molecular biology. Now, each nucleotide, each of these small units is made up of 3 parts: the nitrogenous base, okay? We have 4 nitrogenous bases. And then we have the sugar, number 2 here. We have the sugar, let's point it out. Deoxyribose, it's a pentose sugar containing 5 carbons. And then we have the negatively charged phosphate group. So this will become clear. All right, guys. So this is the structure of one nucleotide present in DNA. So we have a phosphate group here. Phosphate groups are negatively charged. Okay, and this is what gives DNA its negative charged. Okay, this is part of the backbone, of course, of DNA. Then we have the sugar, the pentose sugar, deoxyribose. Deoxyribose, of course, deoxy because it's missing the hydroxyl. Okay, if there's a hydroxyl OH group here in number 2 carbon, then that would be ribose, which is present in RNA. And then we have the nitrogenous base. Okay, we have 4 different nitrogenous bases. But however, in this picture, you guys can only see thymine. Okay, only thymine, but it can be guanine, cytosine, or adenine. All right, now let's look at the DNA structure. There are 4 types of nitrogenous bases. 4 types of nitrogenous bases that fall into 2 families. The first family are the purines. So you can see here the purines right here. Let's have a look at them. These are the purines right here. They are adenine and they are guanine. And they have a 6-membered rings fused to a 5-membered ring. So 6-membered ring fused to a 5-membered ring right here. These are the purines, and they are larger than the pyrimidines. Then we have the pyrimidines, okay, and they are cytosine, thymine, and uracil. Uracil would be for RNA, of course. Okay, and these are the pyrimidines right here. This is thymine. They are 1 ring. Okay, they are 6-membered ring, cytosine, and uracil. Now, as you guys know, DNA is a double helix. Okay, it's a double helix, and here the rungs of the ladder, you have complementary base pairing. So the G pairs with the C. Okay, you have 3 hydrogen bonds in here. The A and the T pair with each other. They base pair with each other. So adenine with thymine, they base pair with each other. Okay, these are complementary base pairing. Okay, so the DNA structure, it's pretty long. It's about 2 meters, and it needs to fit inside the nucleus, which is very small. So how are we going to do that? So how it is done is that the DNA is wrapped, okay, the DNA string is wrapped around histones. These are proteins, beads on a string. They are wrapped around histones, and then they form compact structures. So there are different levels of packing until they become very compact, which is the chromosome form, and then they're able to fit inside the nucleus, okay, because of the presence of histones to which DNA is wrapped around them. All right, guys. So for experiment number 5, we're going to extract DNA molecules. Specifically, we're going to extract DNA from blood. And as you guys know, in blood, there are 3 different types of cells. Only one type of cell is nucleated and has DNA. So this DNA is not going to come from red blood cells or platelets because they both lack DNA. They lack a nucleus and they lack DNA. So the DNA will only come from white blood cells. Will come from white blood cells. So the goal for today, the aim for today, is to break the cell open, to lyse the cell. So we're going to break this cell membrane right here. This is the cell membrane containing the phospholipid bilayer. The nuclear envelope also needs to be broken. It also contains the nuclear envelope, a phospholipid bilayer. So we're going to be using the same method to break this cell membrane. After we break the cell membrane and the nuclear envelope, DNA is released, and then it is purified, and then we can do different analysis with this pure DNA. So this is the aim for today, guys, is to get purified, pure DNA. We want to get pure DNA so we can use it for PCR, for diagnosis, for genetic tests, to study diseases, etc. Okay, so we have to use innovative molecular biology techniques. So what can we do with DNA? What are the applications of the basic molecular biology techniques that you guys will learn in this lab and also in the next lab? Okay, these techniques, guys, are very important, very essential. Why is this? Okay, because they help us in the study of genetic diseases, okay, the causes of diseases, and for the development of drugs, therapeutic drugs against cancer, against Alzheimer's disease, etc. For example, they are helpful in crime scenes, in forensics, in fingerprinting, and for genetic engineering and cloning, gene therapy, etc., and cloning of things. All right, guys. So let's say, guys, that we're going to extract DNA from blood. You have blood in front of you. We got it from the hospital. We want to determine, our goal for today is we want to determine which patient has leukemia, okay? Which patient has a type of leukemia called CML, okay? So the first thing we need to do is we need to extract the DNA from this patient's blood. So to do that, the first step would be to obtain the blood sample from the patient. Let's say this has already been done. I have provided you guys with the blood if you were to do this lab. And then what we're going to be doing today is we're going to be extracting and isolating DNA from the blood. That will be the goal for today, of course, if we are doing the experiment. And then for the next experiment, experiment number 6, we're going to check for the presence of the cancer gene. So this is the abnormal CML type leukemia, okay? So we're going to use PCR. We'll learn about PCR in the next class to amplify this target gene. Okay, so we're going to be using PCR to amplify the target gene to see if it is present or not. Okay, to see if it is present or not. Remember, guys, this is a hypothetical example. Okay, we're just doing this as an example. Okay, it's more complicated than that. This is oversimplification. Okay, so after we get the DNA today, after we get the DNA here, see here, after we get the DNA, next class, we're going to use PCR to amplify the target gene, which is the leukemia gene. And then we're going to be using a technique called gel electrophoresis to see this DNA. That will be next lab. Okay, so we amplified it. Now we have to see it. And then we're going to determine, make conclusions, which patients have this leukemia gene, the BCR, not the PCR, the BCR ABL abnormal gene. Now, the kit that you guys will be using to extract DNA is called the Ultra Clean Blood Spin DNA Isolation Kit. So this is the kit that you guys are going to be using, and you can see here there are many buffers, and I'll explain to you guys what these buffers are. And there's a protein in here. Okay, there are many buffers that are used for DNA extraction, specifically from blood. Now, the basic protocol of DNA extraction, so this is the general protocol for DNA extraction, whether it's from blood, whether it's from a plant like a banana, etc. There are 4 steps. 4 steps are followed to extract and isolate and to get purified, clean DNA from the rest of the cell. The first step is lysis. Lysis basically means, guys, here, breaking the cell open. So breaking it open to release the content. Then we have precipitation. So precipitation of DNA. Usually people add ethanol to precipitate the DNA. In the kit, we have a DNA binding step. We have a DNA binding step. And I'll show you guys here where the DNA binds to a column, binds to a filter. In the third step, we have the washing. Okay, we wash the DNA clean. Okay, make sure it doesn't have any salts, etc. And then we resuspend the DNA. So we add like a buffer or something like that so we can use it. In the kit, we have a similar procedure called elution. Elution is basically where we remove the DNA from the filter. We remove the DNA, the clean DNA from the filter, based on charge. I'll explain later on so we can use it. Okay, so let's talk about the first step, which is the lysis part. So lysis means breaking the cell open. Okay, breaking the cell, like hemolysis in the last experiment, which is breaking the red blood cells open. So lysis means breaking, open, okay, rupturing. So if this was a plant cell, let's say it's a banana, it's a strawberry, etc., we have to use mechanical breakage. Why is that? Because they have cell walls, because they have an additional layer, which is a cell wall. Okay, and as you guys know, in plant, it's made of cellulose, and in bacteria, of course, it's made of peptidoglycan. So we have to use mechanical force, and this means we have to, use, for example, a mortar and a pestle, like break it open with physical force. There are several tools that we can do that with. But if it doesn't have a cell wall, okay, then we move on immediately to chemical disruption. Okay, chemical disruption, which is used for all cells because all cells, they have a plasma and nuclear membranes. Okay, so the cellular membrane is present in all cells, and this is done by adding detergents. Okay, by adding detergents, and you guys learned last class that detergent can break the cell open by forming what's called a mixed micelle, by forming micelle structures, these monolayer structures. Okay, this will create holes, and then the cell will rupture. So after that, what happens is that after the lysis step, that the cells are spilled, the content of the cell is spilled out and extracted into the solution. Okay, so to summarize this, if it has a cell wall, we have to start with the mechanical breakage. If it doesn't have a cell wall, like in our experiment, we go on immediately to the chemical disruption by adding detergent. And at the end of this lysis part, you have the guts of the cell being spilled out. It's not pure yet. Okay? It is not pure. We still have to purify it, but at least we got it out of the cell. All right, guys. So now, as I said, the cell content has been released into the solution. In this cell content, after you mechanically break it or add detergent, contains not only DNA, which is what we want, of course, but also proteins, carbohydrates, etc. We don't want all of these. We don't want the carbs. We don't want the proteins. This is, I consider them garbage in this case. We only want DNA, purified DNA. So what we do is we use a spin filter, what's called a spin filter or a column. And this filter right here, this is the filter. This is the column, sorry. This is the column, and let's move here. So this is the column, and this white thing is the filter. So how does this filter work? Okay, as you guys know, DNA is negatively charged right here. DNA is negatively charged because of the phosphate. Inside the filter, you have positive charges. So these are the positive charges which attract the negative charges in the DNA. Okay, so the DNA which is negatively charged is attracted to the positive charges inside the filter. Therefore, it is stuck to the column. It is stuck to the column, and the other things, the carbs, etc., they are moved out. They move down here, and we have the DNA is stuck to the filter. Okay, so we still need to get it out of the filter, but at least now we have gotten DNA by itself. All right, guys. So how does this kit work? As I said, we need to purify this DNA. It's still stuck to the filter. So let us just go back a little bit. So DNA extraction using this kit, it has 4 steps. 4 steps if it doesn't have a cell wall. Okay, if your cells don't have a cell wall, we go with 4 steps. If it has a cell wall, we go for the 5th step. So the first step, if it has a cell wall, like it's a plant tissue, that would be mechanical breakage. So let's say here, let's move back here. Let's say this is here a cell, a plant cell. Okay, mechanical breakage, like you're grinding here to break the cell wall. But if it doesn't have a cell wall, then we move on quickly. We add detergent. Okay, we add detergent, and as you guys can see here, when you add detergent, the cell content is spilled out. So this is DNA is being released, but you also have here other garbage. It can be carbs, it can be lipids, etc. Then we bind into the column based on charges. So DNA is negatively charged, the column is positively charged. This is called DNA binding. And then we have washing. So I showed you guys the buffers there. Two of the solutions are buffers. And what the buffers do is that when you add them, they interfere with the charges on the column. And so, if there are contaminants stuck to the filter, like the garbage, they get removed, they got washed away. So we get pure DNA. Because even if DNA is stuck in here, you have other garbage that is, so-called garbage that is present. So when you add the washing buffer, it removes the garbage down, and you can throw this away. The last step is the elution part. So the last step is the elution part, and in the elution part, what you have is you add a buffer so that it releases DNA, okay, interferes with the charges here, and it releases your purified, clean DNA from the column. Okay, it disrupts the positive and the negative charges and releases the DNA. This is called elution. And then you can take this DNA and then you can do your analysis with it. You can do PCR or you can do other analysis. So now we have purified DNA. All right, guys. So this is a summary of this kit, how it works in summary. So these are what I'm showing you guys here are the lysis steps. So here we have the lysis steps where we add the detergent. And also we add proteinase K. Proteinase K is an enzyme that breaks proteins. So protein, an enzyme that breaks proteins because we don't need proteins. So this is part of the lysis step. Okay, so it involves mainly using detergent and another enzyme to break proteins. Then we have the precipitation or the binding step. So we have the binding step where the DNA is bound to the column. Then we take this everything, we take the cell content and we add it to the column. So we have the DNA is bound to the column, and this involves the use of ethanol. So if we're doing a precipitation step, DNA would precipitate under ethanol, but in this case, we are binding it to the column. And then we have the washing step. There are 2 buffers here, buffers B3 and 4, that wash the DNA in the column. Okay, because even though DNA is bound to the column, but it might also have other contaminants. So it might have salts, etc. So we need to wash the DNA on the column so that we remove any contamination. And then the last step is the elution step, where we add a buffer that removes the DNA, the purified DNA from the column into the tube so we can use the tube. Okay. In some procedures, we use a resuspension step. This is not that important. Resuspension step, we take the pellet and resuspend it. But in our case, what we do in elution, the final step, elute with buffer B5, is basically from the column, you add this buffer, and basically it takes the DNA out from the column down into the tube, and then you take the tube for further analysis. So don't worry about this slide, guys. If we were doing the lab, we would go over it, but since we're not doing it, we're just gonna skip it. So it's important for us to learn how to use a centrifuge. If we proceeded with this experiment, you would have used a centrifuge, so that would have been more relevant, but let's talk about the background. So if we don't use the centrifuge properly, it will start making sounds. It will get damaged. So to avoid that, we need to balance tubes, test tubes, Eppendorf tubes. This microcentrifuge is for microtubes, for Eppendorf tubes, okay? As you guys can see here inside. So we need to do two types of balancing. The first type is based on volume, and the second type is based on placement, how we place them in the centrifuge. So let's examine first of all how we can balance by volume. So we cannot put our tube by itself in the microcentrifuge. We have to balance it with another tube, preferably with the same type of liquid. And this liquid, okay, these two samples, they have to be balanced by volume. So you can see here, guys, these two tubes are balanced because we have the same volume. Let's say, 100 microliters and 100 microliter here of water. Okay, so this tube is balanced. These tubes are balanced. However, here on the right, these tubes are not balanced because here 300 microliter for, let's say 300 microliters, and here we have maybe 20 microliters. So if we put this in the machine, this will damage the machine, and because these tubes are not balanced by volume. So how we place them in the centrifuge? So notice here, guys, here the orange tube is balanced against this tube. They're opposite to each other. They have to be opposite to each other. And then we have the green tubes. They are well distributed in the centrifuge. So in this case, our tubes are balanced both in volume and in space. So we can proceed and use the centrifuge. Now finally here, guys, let's say we isolated our DNA. Okay, we use the column. We want to determine if the patient has the CML type of leukemia. So we took the blood and we got pure DNA. Now there are factors that can affect the quality of this DNA, okay? Because DNA can break down. So we need to avoid these things, or we need to take care of these things in order, okay, to get good quality of DNA. So if we keep the DNA at high temperatures, this will damage the DNA. Okay, high temperatures can damage the DNA. So whenever you're doing experiments with DNA, it's important to keep your DNA or tissue sample on ice. Okay, you can't leave them on room temperature, otherwise they will be degraded. Now, there are bad enzymes. Okay, bad enzymes called DNases. DNases, this is contamination. Okay, DNases are enzymes that can degrade DNA, can kill DNA. As you guys saw earlier, I mentioned proteinase K. Okay, proteinase K is an enzyme that degrades, kills proteins. DNases are enzymes that kill DNA. Okay, they kill DNA, so we have to avoid these DNases. And how to do that? Okay, we can autoclave tools. So our tubes, our glassware, we can put them at high temperature. We can put them in the autoclave machine, which is the type of sterilizer that kills these DNases at high temperatures. Okay, before we use our tubes, before we use our glassware, we autoclave them. We can also wear gloves in case these enzymes are present, these bad enzymes are present on our bare hands. And what we can also do is we can add EDTA to our solutions. So EDTA is a chemical that interferes with the function of DNases. So we put the EDTA in our sample, and this interferes with the DNases. It kills the DNases. Okay, it takes an important factor in the function of DNases, so it does not allow it to function properly. So we can add EDTA to our sample, and this will inhibit DNase activity. Okay, another factor is excessive mechanical force. Excessive mechanical force, if for example you vortex or you shake the DNA aggressively, this will shear the DNA. This will destroy the DNA. So these are factors that can negatively affect the DNA, affect it in a bad way. Okay, so if you want to get good quality DNA, good quality DNA that is good to use for subsequent experiments, then you need to take these into consideration. So that's it, guys, for experiment number 5. Thank you for listening. If you guys have any further questions, please send me an email.